Speaker should be on. And am I on mute or I'm good?
Yeah, you're good.
Okay, I'm just setting up the, for whatever reason, we have the acquire fast. All right, where scope is in, I can't get rid of this one dialog. Maybe I can minimize it. Maybe that is a little tiny X. There we go. Okay. So I'm going to flip now to the shared screen. We'll talk about five more genera, obscure, rare, uncommon ones in the East, and I think pretty much everywhere. We'll deal with the Anthophorula Expomalopsis group, and we'll then tackle the last of the halictids, Duforea, Diunomia, Nomia groups. And we'll start with Diunomia.
Oh, real quick thing on the, we did have some suggestions. Forgot that I got this email last time. That's my ask. Suggestions for some Western bees, which we don't have a lot of. So I'm wondering if somebody could send us some and then we could cover.
We do have, so sadly we don't have access to the national collection right now. I do have Suzanne Batra's cabinet that she had for years in her house, it turns out. And in there are a bunch of specimens from around the world. I'll try and scroll through that, but with a couple days in advance and see if we can pull out some of the Western genera out of there. So I believe that we should find at least a few and we'll cover those. So yes, I forgot about that too.
All right, proceed.
Okay, so we're looking at a Diunomia male, and they're pretty, in fact, the first time I saw one of these Diunomia males, I actually thought it was a wasp because it was just so strangely configured. You'll see several angles on this. It's particularly the flanges that Nomia and Diunomia have on their legs that really are very, very different and wonderful, but quite different from the normal groups of bees. I do want to show you a problem that is here in terms of the basal vein arching thing. So under I think Michener's key, the McGinley and Danforth Genera Guide, Diunomia would come out as having a strongly arched basal vein, but this is the basal vein of a Diunomia, and the Nomia is pretty much the same. They're in the same subgenus. And you can see it's hard to really make that into a strongly arched basal vein. So this is the basal vein here. So we have it scored for both in the Discover Life Guide. So but it's not a good separation for this particular species. What is useful is, and this is a very large species. This is Heteropoda, which is probably in the East the most commonly run across one. It's a sand specialist. Perhaps all of them are sand specialists. I'm not quite sure. I'm going to fox this a little bit so you can see what you're looking for in both Nomia and Diunomia, if you have any questions, because there's a lot of other features that work fairly well in terms of a gestalt, is that the tegula, instead of being oval, is got projections inward. This is not showing well because it's so dark. Let's see if I can change the light a little bit, get that cursor off of that area. So you can see that it's bumped out here, and it's just overall not an oval shape. Let's see if we can see the other. This is in the males and the females. We're looking at, again, a male now. So you can see here a little bit better, I think, that this is just not a oval shape to it. So and it's also quite large comparatively. The other Halictus and Lazyglossum and those, and pretty much all the other bees, would have a shield over the plate here about half the size. So one has to wonder, like, what's the purpose for that? And that's unknown to me, but it is quite large, and there's several other genera in the world that have these really expanded tegulas. I always think of the tegula as a protection of the base of the wings, but I could be wrong. I don't know if they serve other purposes. Anyway, this is partially diagnostic. And when you look at the abdomen, you will not see what we will see in Nomia, which is very glossy mother of pearl bands of integument, very beautiful across the, I think the apical rims of the tergites here. It's all dark. I'm not sure if all the Diunomia species have dark abdomens like Heteropoda, but they certainly don't have those pearly bands. Heteropoda, again, not sure if it's specific to the species. Very dark wings with even darker tips to them. They're very noticeable in the field. If you see this somewhat wasp-like, these are very elongated wings compared to other species with this two-tone but overall very dark wings, often with an amber tint to them. Both Nomia and Diunomia have modified hind tibias in the males, probably having to do with grasping females, I would suppose, in the males. Here, I don't know if it's just the hind tibia, but it's possibly that the middle tibia has things too. These are species level diagnostic features in terms of the shapes and the sizes of these things. But you can see here femur, tibia, and then the normal tibia would just come down like a simple oblong bead-like segment. But here you can see this giant flange. In fact, you can't even see part of it because it's hidden by the hairs on the other one. But look how large that flange is. That is really obvious in the males.
Can you go back taking a picture?
Okay, let's see if the other side has a clearer shot of that. Or not. Well, it does have a shot. Let's move it slightly here. And we'll look
While
at
you're looking for it, Sam, the geographic origin of this specimen, is this, from around here?
Yes, this one is, so they're very uncommon in parts of the east. This is, I believe, at the northern edge, although I wouldn't be at all surprised that they're in New Jersey in their sand area. So here's the flange again off this side. These were from Maryland, in fact, very close to my house, where I live along the Patuxent River. There's sand mines nearby, and this indeed was in a sand mine section. I even have them coming into my yard, he says proudly, and feeding on a variety of sunflowery things. That seems to be their food plant of choice. And we also found another species, the only place in Maryland, and no state, even adjacent state, has Diunomia, I think it's nevadensis, there too, also in these sand mines. Sand mines in general are very underrated as habitat because we tend to let everything go back to trees. These old sand mines, which were not, let's say, fixed back up by putting a clay cap and sowing crappy non-native orchard grass and trees on them, not that I'm making judgments, are the best thing is to just walk away from them, and they serve as habitat for quite a number of years of these open sand-loving things that often dial out.
And is the flange male specific?
Yeah, these are not in the females. We'll show one in a second. Let's jump to the memorization script here and make sure I'm on. I think Diunomia is the first group here. Yeah. So let's go to the male here and see if there's. So 3 submarginal cells. We don't really need to look at that. Brown to black in presentation of the integument. Tegula asymmetrical, not oval, but elongate with obvious projection or lobe. We saw that. And the rims of the tergites not strongly colored. That separates it from Nomia, normally dark brown or black, with dense edging of hairs, often forming along the rims. I don't know if you noticed that, but that would be true. And then with the obvious and large flattened flange on the rear tibia. really to. I can think, other than Nomia, there's nothing else that will have that large flange on it. Okay, so we saw those characters, and I'm going to jump to the female here, which is very similar in presentation. Oops, let me put my tags back onto the specimen lest they be lost. And mixed up. Okay, that is back on. Here we are again with the female. Another heteropoda, and very large bee, this one, but other species are not nearly as big. If we zoom in, we can see the tegula shape is also very clearly asymmetrical and flanged. Maybe the other side is a little bit better. It's a very dirty specimen. And so here's the shape of the tegula, comes around, and then here's this big kick out on the scutum side there, showing the asymmetry of that. Let's see if there's anything else here that helps. Okay.
Heteropoda?
Yes. And then, well, separating it from Nomia again does not have any of the pearlescent, as they would call it, bands along the rim, and everything is similar, dark edging along the abdomen of bands of hairs, and that's pretty much it. So the asymmetrical tegula, along with the lack of the grossly asymmetrical tegula, along with the lack of a pearlescent band, gets you to this species group. I've got a, do I have it here? Diunomia, I think this is the nevadensis that, just to show you a size difference here, in this specimen, this was the one I believe that I caught, first one caught at in Maryland. Yeah. And if we look at this different specimen, so in Maryland, there's only a handful of records of Diunomias at all, and they're all heteropoda except one. So it's, in general, we're looking here now at the tegula, different color. It is hard to see. Let's zoom in a little bit, but it's got, it's also asymmetrical, not as clear here as, you can see this edge cuts in and then kicks out, runs around like that. This is a female, and so there's no flanges to look at. In this case, looking at the abdomen, it does have light-colored hair bands. So, but the integument is dark, and it doesn't have the pearl bands that we'll see in Nomia. So, jumping now, but that is a good 1/3 the size of that heteropoda. So
Tim tells us that out west, this specimen has a more red abdomen.
Oh, okay. Yeah,
all some if you got them.
Pardon?
Helen Tim, welcome to send us some.
Yeah.
Show them off.
The
or pictures.
Let's see. I'm going to find my Nomia box here. So, and a lot of things that have these nationwide or continent-wide patterns brings up questions about whether there are more than one species, or at least how do you treat things that have such a continent-wide 3,000-mile distribution in terms of differences in their little molecules, particularly because a lot of them have disjunct populations in the east. All right, here's Nomia. So you only really need to see the presence of these bands to know it's Nomia. Nothing else, no other genus has these. I am thinking that there is one species, though, out west, not in the east, which, again, in the east, these are extremely rare, mostly southern, 2 species to the south. And another species, I believe, sneaks in from the prairies. But I believe there might be one, I'm not 100% sure, someone can let me know, species in the west that has all black bands, but I'm not sure if that's true or not. Just coming to mind. Here's the asymmetrical version of the tegula in Nomia going on, and you can see from the wing venation, I get a good angle on it here, that the markings here are more arched. I believe we also with Nomia, just like with Diunomia, have characterized this as both arched and non-arched, but the pearlescent bands are just a dead giveaway. So you can see there's more of an end arch like there are in the other groups of Halictidae in this specimen of Nomia. And if we look at, I believe that, so is this a male? Let's see, let's look. So the males in this group also have flanges on the hind tibia, different shapes and things going on there. I'm not getting a good view of it on that side. Let's shift to this side. Or am I looking? No, here. Or is that on the femur? Was it on the femur last time? This strange. I'd have to look there. Let me flip it back over. I'm going to look at it under the microscope myself here for a second. Oh yeah, there we go. It is on the tibia, but it was a difficult view there to see. So let's change the view. The architecture is a little different, but the idea is the same. And then when you get to species ID in the males of Nomia, you will deal with what that shape is. So here is the basotarsus. I was confusing them in that last one. And this is the tibia, and here is this huge flange coming off the bottom. Again, probably having to do with female clasping or something along those lines. That changes in shape from species to species. So there's the male, and the female is equally identifiable by the presence of that banding.
What Nomia species are we on?
Let's see. That last one was Nomia. These are Western. It says Chilibiata. Now, these are so old that I would want to look up the name to make sure it's not a synonym now, which, because I don't recall that one.
Yeah, it's much
as a name.
one.
Yeah, this one is Nomia in iridescens, which again doesn't ring a bell. But you know, people can look up. Do I have that up? Just to show you again where you can find synonyms. So if you go to Discover Life and you go to the, let's see if I have it open, Apoidea page, no, B Genera guide, no, but if you go to the species page that largely John Asher runs, it has the awesome tool. So just go to bee species. So here you can look up bee species by country and a number of other categories. That's really useful. But if you go to, if you click on checklist, it brings up all the species all in one page. But the cool thing, it has all the synonyms here. So you can do a simple find in there for a name or even just a last name, and you can look for it and determine whether it's been synonymized in one place. So extremely useful, and I use it quite regularly. Anyway, back to our specimen on hand. Here's this pearlescent band, but in this case it's more yellow in this particular specimen, possibly due to age. But if you look at it closely, it has the color shimmers from blue to yellow in there. A couple of the other rims are partially hidden, so we're not seeing it. You can see bits of it there. So that is some of the easiest ones we have to deal with. So if we go now to Duforea, which is also a halictid, again, very uncommon. And I gotta find the specimens. Here we are. So Duforea, I believe, are, I've got to put the tags back on. The Diunomia I took off, so that does not get lost. Okay. Duforea in the east, pretty uncommon. I think there's one on Duforea nova angli, is I believe on pickerelweed, and then there's a Monarda specialist, and there might be another northern one. I don't think there's any southern ones. And the Monarda specialist, I mean not the Monarda, the one that was on Pontidaria was recorded, for example, from the DC area, but I spent a lot of time looking on pickerelweed and have yet to locate it. And if we go back,
Species name?
Yeah. Sorry?
Species name?
Oh, this one, we don't even have any specimens, I think, in our collection. So this one's Duforea, let's see, I'm trying to read this handwriting, Calicaste, something like that, Calicaste.
Sure.
Pardon?
I will start with that.
Okay. Yep. And so let's go to Duforea here.
Are the flanges flattened tibial spurs, or are they projections in addition to the tibial spurs?
That is a good question. I am going to say, just based on what it looks like, that it's a in addition to tibial spurs. So I'm going to guess, though I didn't look, that it has tibial spurs. Those flanges are quite well up the segment versus like the tibial spurs are almost always right at the very apical ends. And I think have a sort of like an attachment point versus just look like some kind of growth. I could be wrong, but that would be my impression on looking at that. Okay, Duforea here, another halictid, as we mentioned, uncommon. So in looking at this, let's see, am I looking at a male or a female here? Let's flip back to here. Appears to be a female. Yes, I would say, based on the scopal hairs. And if we go back here again, so there's 2 submarginal cells, which makes it is the less common state than 3. And the thing that if you see this would always lead you to suspecting that you have Duforea, is the antennal bases are set well below the middle of the face. So we'll see that in both the male and female I have pulled out. And they're separated from the clypeus by not much more than the diameter of an antennal socket. Okay, so the clypeus is short and very wide because of that. and or in addition to that, I should say, and the clypeus upper margin not really arched up into the rest of the face. Scopal hairs on the femur versus on the tibia, and labrum nearly as long as clypeus, although labrum is often hard to see when the mandibles are closed. Okay, let's see what we can see here in this specimen. We look at our wing veins first. Jump over here just to confirm that there are two. A little difficult to see at that angle, so let's change the angle. And indeed, we have two submarginal cells there. Now, when I get a specimen, particularly if it's not immediately obvious, usually I'm looking at the head and the shape of the head, and as well as color and aspects of the thorax and abdomen and sides and legs, all kind of rotating through very quickly rather than isolating one of the features to look at. And so a quick perusal would tell you that, oh, this face is very different in look and feel than other specimens that you would see from day to day. So here's the face, again, dirty old specimen. Head throughout there, the roughly halfway through would be there, and here's the antennal segments quite a distance below. We'll zoom in a bit. Most Duforea, I think maybe all, are quite small, you know, certainly smaller than honeybees. Here's the antennal socket, there and there. Here's the clypeal rim. You can see how it's not very wide or not very long longitudinally, but latitudinally quite wide, and the distance between that suture line and the antennal socket is less than the diameter of that antennal socket.
Yeah, I want to take a picture.
Okay. Okay. And we'll do this again on the other specimen, but no guarantees. Let's tilt this head a little bit this direction, which I think will be a better, more at right angles shot for this thing. So you can really see, let me see if I can get the white balance and lighting a little bit better, but you can really see here's the head, here's the halfway point, how far down the antennal bases are on that head. So let's see, what else did we have? I'm trying to remember.
Did you want to show us the labrum?
Oh yeah, let's see if we can see that. So clypeus short, scopal hairs, femur. Okay, labrum is long. All right, go back. Let's go back to the head here. So the labrum may not be at all visible here. On this specimen, but let's see. Well, you can partially see it. So the mandibles are projecting out here. The labrum, here's the clypeus, and most species other than Megachilidae have relatively wide versus long labrum labrums. But in this one, I can't, it's incomplete. I can't quite see it here. I believe that's the rim of the clypeus right there, almost certainly the rim. And then here's the labrum. These mandibles block us from really getting a clear view of that. So equivocal as to what's going on there. If we look at the hind leg and look at the scopal hairs, theoretically they are on the femur. or maybe it's just that they have them on their femur rather than only, I don't know if we should maybe modify that, if it's got them on the tibia and the femur. I think it would be very unusual to just be on the, yeah. So if we zoom in, we should change that to be on the, so here's the femur. It does have scopal hairs there, but it also has them on the tibia, as would most. So we should modify that to be scopal hairs on femur as well as tibia. And instead of implying, as I kind of saw, that it would be only on the tibia or on the femur. Okay, here's a male, and males tend to have weird looking antennae. I don't know enough about the Western species to know any details. So here's our face again, about how these various long and sometimes kinky and with cutouts in them antennae work in terms of ID. It doesn't appear in the books to be part of the ID string, but again, a noticeable feature. So here we have the base of the antennal segment's very low on the face. And if we got a zoom in on here, you'll see that the clypeus is again very short and wide, although it's got quite a bit of hair on it. And if we can get closer, I'm not sure we can see these distances super well. Here's the edge of the clypeus. There's an antennal base, and the distance is hard to tell here, but it's probably about 1. But normally you don't have to do the check of the antennal base, is just the fact that it's below the head would be the key factor. And yeah, a little bit difficult to say. This does look a little bit slightly wider than the antennal bases, so perhaps in the males it's not as important. Let's see what the guide here says. Well below middle of the face, not separated from clypeus by not much more. Okay, so it is clearly a little bit more than an antennal socket. Clypeus short and wide, upper margin not much arched up into the rest of the face. So if you recall, a lot of the other bee species, the clypeus has a strong arch at the top, and this is very flat or very obliquely arched across the whole part. Again, we have the labrum, which we can almost never see, 2 submarginal cells, which will trim down the numbers of specimens, and then tip of the marginal cell laying on the rim on the margin of the wing. So we can take a look at those. There's
do today's halictids have that characteristic space on T6 that does not have the hair?
I don't. Yeah.
As butt crack?
Right. Those are, I think, associated with the algochlorines. Let's take a look in a minute. So here's the margin of the wing. Here's the tip or apex of the marginal cell. And it's not, I would say, right on the margin, but pretty close. This is hard to define here, but there is a vein that runs along the side of that. And so one could really say that it probably is lying on the edge. It's certainly not out here, further out in the cell like many species. I'm not sure what the separation groups are for that, but there must be a couple other species groups that have antennae below the center, like Hylaeus, for example, I think has at least one species that has that characteristic. So let's look at some butt here, and get in there. Isn't that is that in the male and female? It's not a character I use very much, so but is that in both males and females or just in females?
I think it's
in
both.
algochlorides.
There might be kind of a ghost characteristic in the males.
Okay.
But it's definitely a Halictus lucio thing.
Yeah.
Yeah.
So I don't really see anything there. It might be in the guides as a character, probably is somewhere, but it doesn't appear to be in this particular string. Yeah, I don't see any slits. Let's look at the female. And there's more than one way to come to these ID things, as the Discovered Life guides are sort of designed to allow you to create your own paths to IDs, and simply taking what we've written here in terms of the cheat sheets. You know, you can create your own cheat sheets too, so you can add that in. Yeah, I don't see any that cleft at all either. So good observation. Now, let's go to the last pair. And originally, or not that long ago, I should say, Expomalopsis and Anthophorula were considered to be the same thing. And then the Anthophorula were split out of the Expomalopsis group. The males are easy to tell because Anthophorula has yellow face, or at least pale markings, and whereas the Expomalopsis males do not. Now, I need to find the correct box here and find, okay, let's start with these. So these are both really uncommon in the East. I'm sure they are more common in the West. The Expomalopsis group, I think, is essentially tropical in the East, so you can get a couple species from Florida. At least one, both if not one, are probably introductions from the Caribbean. A little bit difficult to tell what the origin story was. It looks like Mitchell in his book misidentified one of the two that he mentions, and it should have been Analis as the correct species ID. It should be in the updated Discover Life guides. Okay, so these are small to tiny. I'm going to show here Anthophorula, a female, and both these groups have, in addition to being small, have incredible amounts of scopal hairs. So really, really fluffy, and I would presume multi-branched hairs on the hind legs. Particularly surprising because of how small they are. So very fluffy, so very different look than, say, a halictid or other sweat bee that might be small. Pardon?
Species?
What am I looking at here? This is this is Anthophorula texana.
Okay. Thank you.
And let's change that white balance and that. So that's characteristic of both. Now, oops, I believe I have found and spotted a problem here. I didn't have time to go back to the literature and try and figure this out, where I reversed the Expomalopsis, the advice on wing marginal cell things for these two, because what we have in the female, and I also didn't mention a characteristic that's in the guide, so we'll spend a little time trying to suss that out if we have that here. So they have 3 submarginal cells, and the head is flattened instead of rounded over, and the scopal hairs, as we mentioned, very dense. Now, the length of stigma much shorter than the length of marginal cell. So I think I have this reversed. So I think the length of the stigma is actually quite long and nearing the length of the marginal cell as it sits on the margin. So if we go here and we look at this, So here's what we're going to be paying attention to. And I'm not sure where I got this. It's this character, because it's not in the Discover Life Guide when I looked it up. There's other characters that we'll jump to. So here's the stigma, and the length here is about or a little bit shorter than the length on the wing. And in Expomalopsis proper, this is a lot smaller. The length of the stigma is a lot smaller compared to this distance here. And you can see this pulls away from the tip quite a bit. So, and now in the females, that was the main ID string for that group. In addition to being small, less than 8 millimeters, which separates it from a bunch of other things. Yeah, we're done there. But if we flip to Discover Life and look at the character, let's see if I can get that bigger on screen in terms of print. How do I do that? It's not Control, maybe Shift plus? Control Shift plus? I'm in Firefox, so maybe, anyway, I'm not going to spend too much time on getting that bigger, but so it mentions that the females are told apart by the basitibial plate. So in Expomalopsis, which is female basitibial plate smaller in direct comparison. Direct comparisons are always problematic, right? Because who has specimens to compare? Appearing flat without a raised margin and with less hair centrally. If this is not the case and there is no central bare area, then there is no transverse carina on T1 near its base. So this is one of those super ugly characters that you put in when you're desperate. So I'm not sure whose guide this came from. It certainly isn't based on experience from myself, because I see these things very rarely. Anthophorula is something that has shown up a couple of times in the East, like hundreds of miles from its nearest records, and it seems to be the one that we were getting seems to be a specialist perhaps on Agellinus, but perhaps not, because we just don't have that much information. So I've only seen like 2 in my career here. Well, now that we're here, let's just take a peek at said basitibial plate and see what's to see.
We'd also like to know, thank you, Laurel, for reminding us that Anthophorula has switched from Halictidae to Apidae.
Oh, you're right. We're now out of Halictidae and in Apidae.
Expomalopsis as well.
Yep. So here is, at highest magnification I can squeak out of here, the basitibial plate of Anthophorula. Now we will review what it says about this. Oh no, that's the wrong one. This is the one in Discover Life Guide. So I think I need to go back and review this, because I think my guidance is lacking there in terms of the females. So in the Discover Life, it's saying female basitibial plate smaller in direct comparison. We'll see if that's true. Appearing flat, okay, did look flat without a raised margin, hard to tell, but did not seem to have a raised margin, and with less hair centrally. It certainly just was showing a less than complete circle. And so again, this is all those characters are poor because it's basing on some kind of experience that you may or may not have with basitibial plates and other characters. And then if that's not the case, and this is the worst part, if you have an if statement in a character, if this is not the case, then there is no central bare area, and there is no central bare area, then there is no transverse carina on T1 near its base. So yes, that's So we will take a look at that T1 here. Let's go back. And so,
give me a second to take a snap of that so that they will have something to compare to.
Okay, so there's a
lots of pictures
Anthophorula tip as magnified. Says no raised margin. Is that a raised margin on the end or not? It's hard to say. It is. If it is, it's clearly not much of one. Let's go see if there's anything what could be called a transverse margin on the edge. So in the east, you largely don't even have to worry about this because the two groups don't seem to overlap at all. But we know that big jumps in distributions can occur simply because we haven't looked very hard, particularly in the south. So there are, for example, records of Anthophorula in Mississippi where Michener worked. So I don't see any kind of thing that I would call a transverse carina. So a carina meaning raised line throughout this area. So it still is in the pocket of Expomalopsis. Now, let's just jump to the female, or I mean of Anthophorula. Let's jump to the female here this is Expomalopsis solini, and see what we can see in terms of that basitibial plate, since now I am curious too. So here is a female of Expomalopsis. You can see very hairy, legged, and take this down a little bit. See if we can get a shot of the basitibial plate. It should be the area over here. Got a lot of burnout from the microphone.
Can you show the erect hairs along the eye margins that separate these two genera from all others in the U.S.?
The eye margins?
That is what Tim asks.
I will look at the eye margins, but I'm not sure what Tim is talking about. Tim, what are you talking about?
You can unmute.
So for the Expomalopsis in Anthophorula, there are a lot of hairs along the eye margin.
Okay, we'll look for that. There are a line of hairs, did you say?
Yes.
Yes.
Okay. We will go there.
Thanks, Tim.
Oh, this might be bent in the wrong way. So let's see what the other leg is doing. See if we can find a basitibial plate somewhere. Okay. Is that the basitibial plate there? There's the hind leg. Got a lot of too much light going on here. Let's see if that can be. A lot of reflectance. Exactly. Is that the basitibial plate there?
It would be.
I'm at a higher magnification.
There you go. Sharpen it up. Weird.
Don't really see. That's possibly where the basitibial plate is. Wow, we are getting bad pictures. Okay, so it should be where the basitibial plate is right there. It might be bare and reflecting a lot. Let's go back to what it's saying here about Expomalopsis. female, basal tibial plate larger in direct comparison, often appearing to have a raised ridge running along its perimeter that encircles an area of denser hair. Wow.
Yeah, but who's scarier than me?
So I am going to say that is pretty ambiguous. Let's see if we can. So I think part of the contrast, and we need to work on our wording there, has to do with maybe the presence in Anthophorula of a bare area versus what appears to be the case here. A little bit tricky with all these light things that I'm going to play with. But I believe this is the basal tibial plate that's basically essentially completely covered in hairs. So the other one had at least a little bit that was not covered in hairs. So is that reliable? I'm not sure, but that would be what we're looking for in differentiating Anthophorula from Expomalopsis. So Tim mentioned, let's jump to that, in Anthophorula to look at the hairs along the, I'm not sure why we're having so much lighting problems here. But so Tim, are you still on there? So here's the head.
They do from the side because the hairs stick straight out.
Okay.
From the eye margin.
All right. So literally their hairs along the eye margin is what we're looking for. Or unless Tim is just making it up. Where did you find that, by the way?
I went to the B course.
Ah, nice. Is that in the guides?
That's in Michener.
Okay. I'm getting some problematic lighting issues right at the moment. But it can be light or dark hairs?
On the bottom edge.
No, it's the top edge.
Oh,
Uh-huh.
okay.
And they're well-spaced, erect hairs that stick straight up.
All right. Possibly here, 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 and that.
Yeah, it's a whole line, the whole margin.
Okay. Let's see. Problem is, this resolution on this level of this microscope may be tricky. And that's both Anthophorula and Expomalopsis?
Yes, that's how you tell those two from everyone else at a quick glance.
And it can be light or dark. So this might be it right here. Does that look like what we're talking about here, Tim? This line right along here?
Yes, that's exactly it. And it's
Okay.
a line of hairs that are separated as individual hairs that stick straight up. So you have to see it, look at them from an angle.
Okay. All right, I think we got it. Cool. That's a good one. I will add that to our list. Or maybe Claire will.
That one's definitely going to need the pointer.
Okay. Do you want the pointer on it, like there?
No, I had it like nice and squared.
Okay.
I might need to.
You would add a pointer.
Picture, yeah.
Okay.
That's a point.
And males and females, I presume, Tim? Let's take a...
Yes, it's in both sexes.
Okay, so we'll go back to Anthophorula, and we'll look at a male. So the males are easier to separate once you know that it's an Expomalopsis or Anthophorula, which is the males have yellow clipiuses in the Anthophorula. It's a little bit hard to see here because of the light. Oh, there you can see the yellow there. Okay, now let's do some practice of looking at that line of upright hairs. And get some focus going on here.
While you're focusing, I'm going to share that Laurel put in the chat that she's found it useful. have found it useful to refer to the bee morphology drawings by Rebecca Andrus Nelson, RAN, while taking this class. They are the black and white pen and ink drawings in Discover Life, and shared the link. So look at the chat if you want that link.
Yeah, those are available when you go to the section where the ID guides are. If you scrolled down, you would get to those, and they will blow up to the size of a page, and you can print them out. And they're pretty useful to have next to the scope as you're learning your way through. All right, I just think it's in our specimens are probably hard to see. I don't see the upright hairs per se. I could see where they would be.
You're looking straight on. If you can flip it to the side.
All right, let's see. I'm looking at the upper edge here, and I don't see it there. But again, we have old specimens, so I'm going to swap this out with a larger male from out west. I think this is Expomalopsis solidaginus. Anyway, they have a black clipius, so super easy at that level. I want to see if we can get another look at this raised raised line along the eyes. We'll oblique it.
I'm going to give us a 2-minute-ish warning.
We're pretty much at the end here. I don't have any more specimens to show, so if there are questions. Ooh,
that's
Tim.
Nice.
There's your hairs. Look at that. Let's see if I can get even more Maggie. All right, nice. Oops.
Is the picture frozen?
Oh, did I hit freeze? Okay, how about now? Do you see my cursor moving, everyone?
Yes.
Okay, so here are those lovely tall. They're quite long and very upright. Nice.
Delightful.
All right, then we have to kind of figure out how to deal with the basitibial plate a little bit better, and then the stigma versus marginal cell thing just all needs to be, we need to be checking that to update the Genera cheat sheets and probably the guide too.
We're in Anthophorula there.
Pardon?
That's an Anthophorula.
No, this is an Expomalopsis male.
Okay.
All right, so any other questions?
There are. Give me a second once I'm naming this file. Clarify what Tim is suggesting. Are we using this line of hairs to distinguish Anthophorula from other genera?
We're using it to separate Anthophorula and Expomalopsis from other species. So it's not genus specific. It's that group, Anthophorula and Expomalopsis. So Expomalopsini, or whatever the subfamily is. And yeah, and then you have to deal with, well, in the males easy, right? Yellow clipius would be Anthophorula, black clipius would be Expomalopsis. The females, it's confusing because I think we may have written some things down incorrectly, or at least not very well defined, regarding basitibial plates and length of stigma to the marginal cell.
Of Expomalopsis.
Pardon?
I don't think we looked at the stigma of the other base.
Oh yeah, let's do that. So here we have a Expomalopsis female. So the wing here, oops, it may have frozen again. Did it freeze?
Yes.
All right, so how about now? It's moved.
You're good.
Okay. So if we go to the wing here and try and get this thing to behave.
For clarity, are we looking for a long stigma in both genera?
No. In Expomalopsis, well, so the confusion is I may have flipped the advice between the two. So in this one, we are looking at a short stigma. I'm going to bring this up a little bit better. A short stigma in comparison to not the whole marginal cell, but the marginal cell as it appears along the margin. So if you look here, the margin, the marginal cell pulls away from the margin right about there. So here's the length of it living on the margin. And then the stigma, the total stigma area here, let me see if I can get some half decent light in here. So this length, by implication, they imply the entire length is quite a bit less than this length here on the margin. So this is less than that. I'll flip to.
We got frozen images.
Oh, is it still frozen?
I'm seeing it. So are you guys seeing the cursor? What part is frozen? Are you seeing cursor movement and not wing movement, or just
we should be looking
at
at.
What point is it frozen for
We
you?
should be looking at a wing right now, and my cursor should be to the left of the wing in the sky.
That is what I am seeing.
All right, it might be an individual. So if you see it, Claire, you're also going through. Oh wow, it turned just black.
We have mixed results. It's working for me. No movement, seeing an eye, no cursor. So all of the above.
All right, so I'm changing specimens. Hopefully people are seeing this, and I'm going to try and get. This is a male. So here, so it's possible it will be a little bit less. This is an Anthophorula, so this is theoretically a longer stigma compared to the marginal cell, where it's about as long. This is the marginal cell on, it's where it lies on the margin before it pulls away. Let's see if I can magnify that a little bit more.
How are you guys doing on the frozen? We don't really know how to help you from here.
Is everyone frozen except us? That seems odd.
Still frozen, frozen.
We have a bunch of frozens. Why?
Frozen, but at different stages of frozen.
So what are people seeing?
Some, we have a few, and then there's a few people who are not frozen. Okay. Very sorry, folks. We now see a wing.
Yeah, I'm moving it right at the moment.
It is being moved.
Oops.
Okay, friends, I am very sorry. Do not know how to help since it is not a consistent problem. Hope that you'll have the opportunity to check the recording for anything you missed.
So Anthophorula long, although in the Genera cheat sheet, I believe this is the opposite. It says Anthophorula short stigma compared to that, but I think it's long. It should be long stigma that's about the same length as the portion of the marginal cell that's on the wing margin. We'll do one more Anthophorula here to see if it also has a long stigma. Okay, there's the wing, very unappealing, bright white. Take that to there. Okay, so stigma, that length, here's the roughly the marginal cell on the margin. This is a little bit less, but about the same length or so as that. That's supposed to be a character that differentiates the two. With Expomalopsis, I'll try and get one more Expomalopsis in here. Having a short one, that is not even an Expomalopsis.
Not sure if Tim saw this. Does Anthophora sensu strictu have the eye hair trait, Tim?
Should not. if it's actual Anthophora.
Yes, was that a typo or an on purpose? I'm not sure if Tim rolled out.
No, that's Anthophorula again. Problem is, all these old specimens are all Expomalopsis. So I know that Pygmaea is so it should have a long.
I don't see Tim still here.
Okay. I would say, by implication of what Tim was saying, that Anthophorula, that Anthophora would have those, he implied that, so here's the stigma, and here's the section on the marginal cell. So this is long, almost as long as that. That's an Anthophorula. I think he implied that only Expomalopsis and Anthophorula had those raised lines, and that everything else was different species.
So you showed us like 4 Anthophorula wings.
Right. So now I think I've got a Expomalopsis here. Problem is this collection is not. This should be a short stigmad jobber. All right, and a lot of Expomalopsis have very shiny butts, but I don't know if that's a good character. So here we have Expomalopsis. This length is much less than the length of this marginal cell on where it attaches to the margin.
Move your cursor. There you go.
Let's see if I can get that color to shift a little bit. All sorts of lovely lighting problems there, but okay. So, other questions?
Just a reminder, there's Anthophorula in Expomalopsini. Pronunciation is fun. Versus Anthophora in Anthophorini. Anthophorini, eeny, meeny, miny, mo. No other questions? You can unmute if you have them. We're all stuck at different screens because it's high traffic time. That is an excellent hypothesis.
Ah, okay.
So the hairs are a diagnostic trait of the Expomalopsines.
Yes.
Science is all about communication. Cool. All right, well, I'm going to stop the recording. We'll be here for a minute or two while that loads. And I took the most pictures. So those will all be in the files. Eventually I'll probably edit them to point at things, but the file names are extremely descriptive. So I think you'll be able to figure it out because you're all very smart people.